Hi, folks. Okay, so we are going to quickly talk about B and T lymphocyte development. We're going to start here with T lymphocytes. Just recall that T cells must be activated to do their jobs. They cannot directly respond to antigen, which is why you need antigen presenting cells in order to present antigen to them. And remember, APCs can be macrophages, dendritic cells, as well as B lymphocytes. Also recall that T lymphocytes can be differentiated into two types specifically, either CD4 positive T cells or CD8 positive T cells. Your CD4s are going to be your helper T cells and certain subsets of those, and your CD8s are going to primarily be your cytotoxic T cells. So as a review, let's remember that T cells arise or they're produced in the bone marrow, right? Primary lymph organ, like all your blood cells, but they migrate, they leave the bone marrow and they migrate to the thymus gland in order to mature. So on the surface of a T cell, you find TCRs, that stands for T cell receptor, that can only recognize antigen bound to the surface molecule MHC. Remember, MHC stands for major histocompatibility complex, and there are two classes, really three, but there are two that you need to be concerned with in this part of your immunity material. So MHC class 1 and MHC class 2. Now, the type of T cells, whether it be a helper T cell or a cytotoxic T cell, is going to bind to either the MHC class 1 or the MHC class 2. So a reminder, CD4 cells, they're going to bind to MHC class 2, and your CD8 cells are going to bind to MHC class 1. Now, when you think of these MHCs, you should really think of a receptor that's found on mainly your antigen presenting cells. And they're capable of producing or holding on to antigens, whether it be a peptide or a glycoprotein, something from the surface cell of a pathogen like a bacterial cell or maybe a viral cell. And these MHC receptors are able to sort of link up with these TCRs on the cell surface of your T cells, and that's where the antigen recognition actually happens. So naive T cells encounter the antigen that's bound to the MHC, the T cell proliferates, remember that means to grow, and then differentiation happens of the T cell itself. So it can differentiate into a memory T cell, right? Or it could differentiate into an effector cell, which is a cell that's able to be activated or it's able to do its actual function, right? And we're going to take a look at what that actually means. And before we do, what we really need to sort of take the time to do is understand that T cells, before they're activated, before they're mature, have to go through a pretty lengthy selection process. It's called central selection. Okay, this happens for all lymphocytes, not just T cells, but B cells as well. B cells go through their selection process in their primary lymphoid organ, which is the bone marrow. T cells, when we said that they move right from the bone marrow to the thymus to mature, well, they actually move there so that they can go through this selection process, okay? And you can think of this positive negative selection process sort of like quality control. It essentially is a series of steps that your immune system takes in order to make sure that your B or T cells, your lymphocytes, are properly structured and properly functioning. Okay, mainly what this means is they want to make sure that your lymphocytes are recognizing foreign antigen, because that's their jobs, right? We want to recognize non-self, but that they aren't reacting to self-antigen. Because remember, we have antigens. Antigen is a general term for something that can possibly be responded to because it's on the surface of the cell. An immunogen, to be immunogenic, is a type of antigen that actually stimulates an immune response, right? We want that to happen for foreign material. We do not want that to happen for self material. So when we talked about blood cells briefly, we talked about how they have a series of surface molecules, right? Certain sugars, for example, and those are technically self-antigens, and they're not something that we want our T cells to respond to and to react to. Why? Because then our T cells are going to destroy our own host cells, and that's how we get autoimmunity and hemolysis of our own red blood cells, as well as other host cells in the body. So these two processes, positive and negative selection, they go in order, typically like this. Usually positive selection happens first, and then negative selection usually happens after that. And really what this is, it's a process of self-tolerance. Okay, we want to make sure that when we send these T cells out into our bodies, they're capable of tolerating all of the host cells and not tolerating any foreign cells. So what I want to do quickly is I just want to kind of bring up a little picture. I already drew to save time. And kind of show you what this looks like. Otherwise, the idea of positive and negative selection can be a little confusing. So recall that we have our bone marrow here, this little picture of a bone, and inside we have our immature developing B cells, T cells, as well as other blood cells, right? Hematopoietic cells. So essentially what happens is we build this T cell, and we know it's not going to mature here because we know that it matures where? In the thymus. So essentially, this T cell is going to make its way to circulation. It's going to move from the circulatory system to the thymus gland. Okay. And inside the thymus gland, you have what's called thymic cells. Okay. It's a type of epithelial or endothelial cell. And it's important to note that those are there because they're going to play a really important function in this whole selection process once this T cell makes it into the thymus gland. All right, so I have a big picture here of my, my thymus gland, and then I have a series here of thymic cells. That's what these guys are. All right. And let's say that this cell right here, right, this was my immature T cell. It's the one that moved from the bone marrow to the blood to the thymus. All right. So this is my starting point. Now, my immature T cell, by the time it gets to this place, this position in the thymus, it's going to have expressed a certain number of cell surface molecules, okay? Tons of them, most of which we're not going to talk about in this class. The ones we are going to talk about are ones that we've kind of already presented in the past. So for example, we have our CD markers, right? And remember, this is an immature T cell. It hasn't differentiated into its specific type yet. It hasn't become a CD4 helper cell or a CD8 cytotoxic T cell. So it's going to express both of those CD markers. So let's say that this is CD8 and this is CD4. Okay, kind of looks like a bunny. It's also going to express the receptor that we talked about just before at that previous slide, TCR, which stands for T cell receptor. Okay, it's going to have a couple of those. So this is our immature cell. It's kind of expressing everything at this point because it hasn't chosen a destination, right? It doesn't know what its job is going to be. So essentially these thymic cells are going to interact with this T cell, all right? Think of this as steps, all right? This is the same T cell. And it's going to put it through a certain number of tests, all right? And those tests are to make sure that the T cell is capable of recognizing the receptors that will present foreign antigen, so anything that's non-self, and then it will test to make sure that this T cell isn't going to recognize self antigens for destruction. It's not going to react to them, all right? So in this first step, let us take a look at this thymic cell. This thymic cell has two complexes, two receptors on it that should be easily recognizable to you. This first one that I drew in green is an MHC class 1 molecule, and the one in blue is an MHC class 2 molecule. Okay, remember, these are antigen presenting molecules found on the surface of our host immune cells, such as macrophages or B cells, and their purpose is to present antigen, right, to T cells that are going to do work on it. But also recall that the MHC1 cell is supposed to react or present to CD8 T cells, and that the MHC2 is supposed to react and present to CD4 T cells, okay? That is a 4, not a 9. So this guy is going to move over to this position. So it can be in front of this thymic cell, right? And when it does that, it is going to line up its CD8 molecule with the MHC1 on the thymic cell, and it's going to line up its CD4 molecule on the MHC2 of the thymic cell, all right? And then, so let's just write that CD8, and remember this is CD4. Again, it looks like a 9. All right. And when it does that, it is checking for one thing in particular, good binding. All right. In this case, we want good binding. All right. We want to know that if this future T cell, if it's going to be a cytotoxic T cell, a CD8, we want to know that it's going to be able to recognize and respond to MHC1 molecules in the actual peripheral blood or in the lymph, whatever, when it's out in the body doing its job, when it's a big T cell. If it's going to be destined to be a CD4 helper cell, we want to know that it's able to recognize and react to MHC2 molecules on the surface of other immune cells. All right. So we're looking for, is there a good fit? All right. Is there good binding? All right. If there is good binding, that's a good thing. Smiley faces all around. Okay, so this is a check mark. We want good binding in this first step. If there is bad binding, okay, or poor binding for whatever reason, maybe there was some issue in the amino acid sequence of this complex, maybe there was some denaturation, for whatever reason, the fit isn't good. Because remember, goodness of fit is important. We talked about that last class. If there's bad binding, well, then this molecule is going to be thrown out. It's going to be signaled for apoptosis, all right? Cell death. Now, because we're looking for good binding here, okay, we're looking for a positive reaction, this first step is what we call positive selection. Positive selection. Okay, that's what we're looking for in positive selection. So back here, positive selection is a double negative T cell exposed to the MHC, all right? T cells whose receptors recognize self-MHC molecules. So positive selection is all about being able to recognize these major histocompatibility complexes, all right? And then cells that fail that selection are going to be eliminated in the thymus, all right? They'll never leave the thymus. They're never going to make it out. They're going to be destroyed via apoptosis, okay? So that's positive selection. So if it does that, okay, if it makes it through that stage, it's going to move on to the next step. The next step, okay, is going to be checking for something else. The next step is essentially going to be checking to see if this T cell, still don't know if it's a CD4 or CD8 yet, but when it gets to its future job, is it going to react with self-antigens, antigens that belong to your body? Because if it does, that's probably going to be a problem, right? That's how you get autoimmune disease and a slew of other things. So in this case, we are going to not be reacting our CD4 and CD8 molecules. In this case, it's going to be the TCRs. Okay, it's going to be the T cell receptors that are specifically sort of reacting with these molecules here. And now you'll see these little red dots here, okay, on these complexes of the thymic cell. Those are self-antigens, okay? They're put there on purpose because that's what we're checking for, reactivity or no reactivity. So when the TCR sort of binds, what are we hoping for? Well, we're hoping that the binding site, the active region of these T cell receptors are not going to have good fit with the self-antigen, that they won't have good binding. All right, so we're hoping that we'll have bad binding, okay, or poor binding. And if we do have poor binding, that means that this cell isn't going to react with our self-antigens. That's a good thing. It can move on next step in the process. So we're happy. All right, that's good. However, if for whatever reason, the active site of these TCRs are able to bind well with the self-antigen molecule, so in that case, we have good binding, that's a bad thing. We don't want that. That's going to lead to issues in the future. This T cell shouldn't be allowed to move forward in the process. Okay, they did not make the cut, and they're going to be flagged for apoptosis in the thymus, and they will not move out of the thymus in the future. Okay, but we're going to assume that we had bad binding here. Okay, and what do we call this? Because we're looking for this sort of bad binding. We call this negative selection. All right, so this step of sort of checking for reactivities to self-antigen is a negative selection. So if they pass the positive selection stage and the negative selection stage, then they move on to the next stage. And what is the next stage? Well, what I have here is I have our T cell that's passed these previous stages, and I have an epithelial cell, a thymic cell up here with the MHC2 molecule, just like before. And then I have another thymic cell here with an MHC1 molecule. Now recall that we said this immature T cell had both CD4 and CD8 on it, okay? All over, expressing in fairly equal quantities across the cell surface. Now, as it's floating through here and it passes this thymic cell and this thymic cell, if it may just so happen to come across this thymic cell, and if the CD4 molecule, all right, if this CD4 molecule just so happens to sort of bump into this thymic cell and bind with its MHC2, guess what? This T cell is now going to be destined to become a CD4 helper T cell by sheer accident, by sheer coincidence, okay? This is sort of a random step, all right? Now, on the other side of that, if this T cell, if its CD8 molecule happened to bump into a thymus cell with an MHC1 complex, and these guys, for even just a second, were able to bind, all right? That's gonna activate this T cell to become a CD8 positive cytotoxic T cell, all right? So again, it's this sort of random step, kind of like when we're talking about transmigration and crossing over during meiosis, okay? So this is a random step here, all right? So once we've established what kind of cell it's going to become, what's gonna happen then? Well, at that point, either one, all right, of these cells are going to leave the thymus. At this point, they are allowed to because they've passed all of the steps, right? And they've had this sort of destination moment where they've decided what kind of cell they're going to be. And they can move to different areas, for example, the lymph node, okay? Or they can go to the spleen, all right? They're going to go to areas, secondary lymph organs mostly, because that's where they're going to come into antigen or come into contact with antigen, all right? So they're going to go and they're going to start doing their jobs. And that's basically what's going to happen. One thing I do want to mention, okay, because it will make it a little clear later, is that there is one more destination pathway for these guys. Both TH cells and TC cells, all right, CD4 and CD8, they're both capable of going through a further differentiation process into another specialized type of T cell that we call a T regulatory cell. Okay, so they can become a T regulatory cells, and T reg cells tend to be considered suppressor cells. They do exactly what they sound like. They actually regulate other T helper cells and other cytotoxic T cells and make sure that they're kind of doing their thing. So they are capable of differentiating into this specialized T regulator cell. Now, one question you might have is, hey, this T cell had CD4 and CD8 on the surface of its cell. How did it just suddenly become a CD4 or a CD8 and that's it? Well, at this step, when it binds, so let's say it was the CD4 that bound to the MHC2 molecule here, what that's going to tell the cell to do is it's going to say, upregulate CD4 molecules, upregulate CD4 molecules on the surface of this cell so that you're going to have just CD4 molecules all over the surface. And it's going to say downregulate CD8 cells. So any CD8 cells that were on this T cell are going to be downregulated, meaning that they're no longer, they're going to decrease in expression on the cell surface over time. And the same thing happens over here, only the opposite, right? If they bind with a CD8 binds with an MHC1 in this random step, then it's going to say to upregulate CD8, right? And it's going to say downregulate CD4, and then you're just going to end up with a bunch of CD8s on here, and that's pretty much it. Okay, and that's how that process works. And that's how we end up getting these really specified T cells with individualized CD markers. They start out with a bunch of the same ones usually, and then they sort of differentiate into only having one type or the other based on the result of this step right here. But before that, positive and negative selection had to happen first. Okay, I hope that made sense to you guys. And of course, if you have any questions, you guys know you can come see me at any time. You can shoot me an email, no biggie. All right, so moving right along here. So clonal selection accounts for most of the properties of the adaptive immune system. This is essentially just kind of covering the bases of what we just talked about. It's saying here that these mature T cells that have survived the selection process, okay, will either become a CD4 or a CD8 positive T cell. That's what it's saying, okay? They're gonna migrate from the thymus, they're gonna go to the medulla of the lymph organ, or they're going to go to the spleen, or they're gonna go right into circulation, okay? But essentially, you're gonna become one or the other, and they have to survive that selection process. Otherwise, they get voted off the island for apoptosis. All right, so T lymph subsets. Now, I kind of mentioned this before, all right, but so essentially what we're talking about here is we're introducing this idea of T helper cells having multiple types, all right? Before this moment, it's always been CD4s are T helper cells and CD8s are cytotoxic T cells, and that's fine. What you need to know is that T helper cells, so CD4 cells, have different differentiation paths as well. So they can further differentiate into a certain type of helper cell. All right, and there's a couple different types. You have you have helper T cell 1, helper T cell 2. You also have a helper T cell 17. All right, so there's different subsets of helper T cells that we can kind of go into depending on what the function of the cell is going to be, or what the function is that's needed, right? So this picture is pretty good because it kind of shows you that all the different destination pathways of these helper T cells. So 0 or not, meaning we're just kind of like at baseline here. So essentially what happens is that you have an uncommitted helper T cell here, right? It hasn't committed to a differentiation state yet, that has left the thymus, right? And it has gone to some secondary lymphoid organ, like we talked about before. And then what does it do? It comes into contact with antigen that's being expressed on the surface of this, antigen presenting cell. Maybe it's a macrophage, maybe it's some sort of dendritic cell or a Langerhans, who knows? And essentially this interaction, the exposure to a certain type of antigen, right, is going to lead this uncommitted helper T cell into one of these pathways. And notice T regulatory cells is on here too, because remember, helper T cells can assume that role as well. So let's say that we go in this direction, we go in this pathway. There's exposure of the uncommitted cell to interleukin 12 and interferon gamma, okay? That's going to destine it to become a Th1, a helper T1 cell, all right? Helper T1 cells are capable of releasing these cytokines, interleukin 2, interferon gamma, as well as tumor necrotic factor alpha, all right? And their jobs are mostly in cell-mediated immunity. Remember, not humoral, but cell-mediated immunity. They deal primarily and respond to primarily intracellular pathogens, okay? So pathogens within the cell, like viruses and bacteria and whatnot, okay? They also are capable of activating macrophages and stimulating B cells to produce IgM. Now, if we go this route, okay, just another possibility, there's exposure to interleukin 4 and interleukin 2, we're going to end up with a helper 2 T cell, all right? They are capable of releasing these cytokines or these interleukins, and they're usually involved in antibody-mediated immunity, which is your humoral immunity, okay, right? Humoral, think fluid, think antibodies floating around in fluid, all review. And they deal mostly with extracellular pathogens like parasites, okay? And also your hypersensitivity reactions, so asthma, allergy, and other types of type 1 hypersensitivity reactions as well. Also, we are going to have the possibility of becoming a helper T17, and it's the same thing, guys. They'll be exposed to these interleukins, this beta TGF, and it's capable now of releasing these cytokines, and it deals with extracellular bacteria, right? So that's mainly in the skin, the lining of the intestines, okay, mucosal surfaces. So that's going to include fungi as well, all right, and it also has a role in autoimmunity, because one thing, the sort of theme of immunology so far is this idea of overlapping jobs. You know, some do the same thing, but there's always one distinct job that kind of separates it from the other as far as subsets of leukocytes or lymphocytes go. Okay, and then finally, we already talked about T regulatory. If it goes down this path, it's involved in immune tolerance, okay? It's going to regulate other helper T cells, other cytotoxic T cells, and it's going to keep everybody in check, okay? Take into consideration that all of these cells, no matter which pathway we go through, they're all going to take that second step and they're going to go through some sort of expansion, okay? They're going to have to proliferate. So we'll end up with more than one of just these Th1 cells or Th2 cells down the line. Okay. So review, same thing, your helper T cells, what do? They orchestrate the adaptive immune response. That's great. They do what they sound like they do, right? They help out. We have different subsets or different possible outcomes that we can get. Th1, 2, 17, and also remember T regulatory is another type of cell that helper T cells can become. And then, of course, the cytotoxic T cells, which are CD8s, we find them in peripheral blood. We also find them in the secondary lymph organs. And the most important thing to remember about them is that they can destroy, cytotoxically, they can destroy host cells that are targeted, okay? Host cells that are infected. That's what your cytotoxic T cells are doing, okay? So if you have a viral infection in your cell, your cytotoxic T cell is going to target that for destruction. Also, tumor cells, right? Any cancer cells, if any of your cells are showing any oncogenic function, or if there's any genetic mutations or defective cells in general, okay? Any of these cells, any problem cells, your cytotoxic T cells can respond to for destruction. All right, but the important thing to remember is that because they were able to succeed or pass that selection process of positive and negative selection, they spare non-targeted or non-infected cells, okay? So this is just a picture of different basic a visual aid, if you will, of functions of helper T1 cells and kind of what they look like depending on which way they go. So it starts out the same way with all of these, right? That we've talked about a million times. We have a pathogen. In this case, we're depicting a bacteria. You have an antigen presenting cell, maybe a macrophage, right? And what does it do? It endocytoses it, okay? And then it phagocytoses, meaning that it's going to destroy, digest the pathogen, take those cell surface antigens, present them through what molecule is this? Well, we're talking about helper T cells here. So this molecule must be an MHC class 2, right? And that's the molecule we're using to present that antigen to our helper T cells, our CD4s. Once that happens, this naive T cell, remember, naive T cell means it has not been exposed to an antigen yet. Once that happens, it could be activated and differentiate into what kind of cell type. Maybe it differentiates into a Th1 cell, right? This one here wasn't on the previous slide, TFH. This actually stands for follicular helper T cell. It's just another T cell type. And then they go on and they do what these T cells do. If it's a Th1, remember, we're talking about intracellular pathogens, right? That's what Th1 tends to do. So it's going to go through the phagocytic process, all right? They're going to release interferon gamma, and then we're going to enhance the pro-inflammatory response. On the other side of that, if we have a helper T cell like this one, that's going to activate a B cell, right? And if it does that, what's the B cell going to do? It's going to go through clonal expansion, right? It's going to proliferate. It's going to differentiate into plasma cells. Those plasma cells are going to release antibodies. And then those antibodies are capable of participating in a myriad of things that help with the pro-inflammatory response, specifically opsonization. So interacting with the FC receptor, right? So the FC receptor of, let's say, this macrophage, this phagocyte, is able to interact with the FC portion of the antibody as it has binded to the surface antigen of, let's say, this bacterial cell. And that's going to say, hey, macrophage, this bacteria is delicious, right? It's going to make it more attractive, and it's going to make the macrophage, it's going to activate it in a way that it has better phagocytic activity. All right, it's going to say, come over here and eat this bacteria. Okay, and complement works the same exact way. So we can also activate which complement pathway. It's going to be classical, because classical is going to be our antibody mediated complement pathway. Same thing here, you have helper T cells, but this is going to be helper T2 cells. And remember, keep in mind that these types of cells are going to be dealing mostly with extracellular pathogens. So that's going to include mainly parasites, because those are the ones that tend to be extracellular. So we have a helminth over here, right? So outside of the actual cell, it's not invading a cell, but it's still capable of being phagocytosed by this APC, this antigen presenting cell. And once it is engulfed, right, the same thing happens. Antigens are presented on what kind of molecule? This is an MHC class 2 molecule, because we're talking about helper cells, right? CD4s. Same process happens, proliferation, differentiation. It can become one of 2 or one of 4 different types of helper T cell subtypes, and then it goes through whatever process it's capable of going through. So in this case, it's the same thing as before. A B cell is going to be activated, right, with antigen. It's going to go through clonal expansion. It's going to make or differentiate into plasma cells. In this case, we're talking about a parasite. So it's going not only to produce its normal IgM, IgG, but it's also going to produce IgE antibodies, because recall that IgG immunoglobulins, they respond to parasitic infections, okay? And they tend to be elevated during parasitic infections, which is why we have a mast cell here, because mast cells, tissue basophils, they respond to parasitic infections as well. That mast cell is going to bind to the IgG antibodies, degranulation is going to happen, and a release of anti-parasitic enzymes are going to go out into the cytoplasm, and it's going to be able to help in the process of killing this parasite. Okay, everything else here, we already know, it's all the same stuff. Eosinophil activation, right? Because eosinophils are the major leukocyte that's involved in parasites, but note that we also said that mast cells and basophils were as well, okay? So that's good, because we actually got to see in this process the release of a different type of antibody from our B cells, so that's nice. And then helper T cells, version 17. we have a bacteria here. And these guys, remember, they deal with fungal infections, but they also deal with bacteria as well. And it's the same type of thing. The one difference with your helper T17 cells is that they majorly release and secrete cytokines of the interleukin 17 variety, as well as interleukin 22. And this is interesting because what interleukin 17 does is it acts on your endothelial cell lining on receptors there, as well as other leukocytes like white blood cells, which is going to then promote those to release their own set of cytokines. So tumor necrosis factor, other types of interleukins, as well as chemotactic agents. And what does this do? It mobilizes, sends signals to neutrophils. Here we have some segmented neutrophils here to come over to the site of infection, right? Which is just going to increase the inflammatory response, and it's going to promote more of the diapedesis into tissue of your leukocytes, more neutrophils, more macrophages. It's going to release certain antimicrobial peptides. This includes complement, and then it creates a barrier around that site of infection so that we don't move out of the tissue. So that's basically what that looks like. And we've talked about this before when we drew that big picture of inflammation in the second line of defense in the innate immune system. So now we're seeing how helper T cells can sort of lend an aid to that process as well. Okay, so CD8 T cell responses. Of course, now we're not talking about helper T cells. We're talking about cytotoxic T cells. And this is more of what we had said before. Here we are this is a lymph node, right? We see a naive T cell. Remember, this is a naive T cell that had matured in the thymus gland, which means it's gone through both positive and negative selection. It's also been selected to either become the CD4 or CD8. In this case, it's a CD8. It's going to enter this lymph tissue, where it is going to be exposed to antigen, right? It's going to be exposed to antigen from an APC, antigen presenting cell. And then once it does that, it is activated. It can leave the lymph tissue. It can enter the circulatory system, right? And where is it going? Well, it's been exposed to this antigen. It's now familiar with it. It's now activated. It's going to start moving to the site of infection. Okay, it's going to move to where the actual infection is occurring, of where this antigen originally came from, right? So once it goes to the site of infection, it is capable of cytotoxic killing. That is its job. All right. How does it do that? Well, it releases things like granzymes and perforins, which we've talked about in the past. It stimulates the release of caspases so that any infected tissues, any infected macrophages or other host cells in that infected area will be flagged for apoptosis, right? We want to destroy those infected cells and essentially death of the initial infecting pathogen. All right, so suppressor T cells, remember, these are just T regulatory cells. We call them suppressor T cells because they're capable of, sort of suppressing the function and action of helper T cells, as well as cytotoxic T cells, and also B cells as well. All right. And they do that by down regulation. Okay. They'll release certain chemicals, certain cytokines that will lead to the down regulation of the activity of these cells, which is good. It's essentially like a referee, someone to keep everyone in check, because once we have activated B cells and activated cytotoxic cells, and there's all this killing happening, sometimes you need someone to come in and say, hey, you can calm down now. All right, we've killed mostly everyone, got a couple more players here, and then we're going to clear, right? Because the end of this active acute inflammation, remember, the goal is clearance. We want to clear all of these immune system cells out of the area, because without that, we're going to lead to chronic inflammation. Chronic inflammation is bad because of all the things we already talked about, like reactive oxygen species and whatnot. Okay. One thing I do want to mention here is that these suppressor cells, or these essentially Treg cells, Many of them are going to be CD8. In fact, the vast majority will be cytotoxic T cells that have differentiated into a suppressor cell, right? But we mentioned before, CD4 helper T cells are also able to differentiate into reg cells or suppressor cells as well. Just they don't do it to the same extent, all right? And it's hypothesized that has something to do with CD marker 25, that it's involved because CD marker 25 is responsible for increasing binding avidity for self-antigens. So that's kind of where the science is going right now, but it hasn't been fully established yet. And right, so CD25, okay, is one of the markers that you find on these suppressor cells, as well as other CD markers. But essentially, you have these Treg cells, right? And they control autoimmunity, right? Because if you have a cytotoxic T cell, that won't stop. Eventually, it might start destroying its own host cells, right? And that's not good. So these CD25 lymphocytes, they occur in the thymus, don't they? We said that a T cell will go through positive and negative selection, right? And then it will be chosen for a destination, either CD4 or CD8. And then after that, some of them will be selected to differentiate further, take another step into a regulatory T cell or a suppressor cell. Okay, so again, 5 to 10% of the total population of these regulatory cells are going to come from CD4 helper T cells, but the vast majority, as you can see, are going to be CD8 cells. Most of those are going to be the ones that are selected to sort of start their future as a suppressor cell. Natural killer cells, remember, these are lymphocytes as well, aren't they? They're type of T cell. However, they are part of the innate system, and I cannot stress this enough because they're non-specific, right? They're capable of doing things that certain T cells are capable of doing with the fact that they don't respond to a specific antigen. All they do is respond to non-self. So if you don't belong here, then we can kill you. That's it. It won't respond or react with self-antigens. Why? Because natural killer cells also go through positive and negative selection. So these are CD5 or CD56 positive cells, right? That's our marker. It's highlighted for a reason. It's important to know. If you want to identify a certain lymphocyte, through some sort of test that we're going to talk about in a second, you're going to look, and if you see CD56 is positively and highly expressed on the surface of this lymphocyte, you're going to say, okay, that's probably a natural killer cell. All right. And that's essentially how that works. So what they do is they're able to kill through extracellular non-phagocytic mechanisms. Okay. It's called a cytotoxic reaction. Sounds familiar, right? And it's MHC unrestricted cytolysis, meaning that you do not need an MHC molecule to present to a natural killer cell. All right. It's not like presenting to a cytotoxic T cell, but the mechanism in which they kill is virtually the same as a cytotoxic T cell, right? It's going to release perforins and granzymes and caspases that are going to sort of penetrate the infected cell, and it's going to lead to apoptosis and lysis of the cell. So it's the same sort of mechanism. It's just how they start is a little different. It's non-specific. So they're mononuclear, all right, just like your other lymphocytes, and they kill target cells that have been sensitized with antibody, right? So when we say that something has been sensitized with antibody, what do we mean? We mean it's been coated, right, with antibody. It's been targeted or flagged by antibody. And it does this by interacting with the FC portion of the antibody, right? So let's recall that on the antibody, right, on the heavy chain of the antibody, we have an FC sort of molecule on the surface of it, and natural killer cells, just like other APCs and macrophages, for example, they have these FC receptors. And so the binding of the FC receptor to the FC portion of the antibody allows for interaction, right? That will eventually, in macrophages case, is going to lead to phagocytosis, but in an NK cells case, it's going to lead to cytotoxic death, just like in a cytotoxic T cell. Okay. So essentially, most of this is just a review of what we've already said. Okay, we've already talked about the fact that CD8 cells are going to bind with MHC1 class 1 molecules, right? We say that they interact with cytoplasmic antigen. Why? Because they're going to interact with antigen or infection inside the cytoplasm of the host cell, right? Cytotoxic T cells are going to destroy host cells that have been targeted for destruction because they're infected, as opposed to CD4 helper T cells, which interact phagolysosomal antigen. Why? Because that's what happens with the macrophage, essentially, with the helper T cell, through that sort of extended interaction, right? You're going to have antigen that is endocytosed. It's going to go into a phagosome, which is just a vesicle holding the actual antigen or the pathogen, whether it be a bacteria or virus. And then that is going to fuse with a vesicle called a lysosome inside of that cell, that phagocytic cell. It's going to expose the enzymes to the bacterial surface so that it can be digested, right? So TCRs, again, react with antigen. TCRs are on all T cells, okay? It stands for T cell receptor. They're on all T cells. Remember, they were a major part of negative selection, okay? APC cells are going to be your antigen presenting cells, and they include dendritic cells, macrophages, B cells, and certain tissue cells. One thing that's new here is endogenous versus exogenous pathway. What's the difference between them? Well, you'll notice that endogenous pathway is associated with our cytotoxic T cells, our CD8s, right? Our CD8s, of course, are associated with MHC1 molecules. This is a little backwards, but that's okay. And then our exogenous pathway is associated with our helper T cells, which is associated with our MHC2 class molecules, right? So what does that mean? So we call the endogenous pathway for CD8 cells, we call it that. This usually has to do with like viruses are a good example of this. So pathogen is processed transcriptionally, right? And the host cell produces antigen through translation before presenting the antigen on the cell surface, right? So essentially think of a virus in the way that viruses will invade your host cell, and then they'll actually incorporate a piece of their antigen, a piece of their own self into your genetic material, right? And what's the purpose of that? Well, the purpose of it for a virus is so that your cells sort of get tricks into reproducing that material, right? So as opposed to making your own proteins, you're now going to make viral proteins, right? So you have essentially the host cell processes this in its own metabolism. That's not what happens with exogenous pathways. With exogenous pathways, we're talking about phagolysosomes. We're engulfing this bacteria, we're keeping it surrounded in this pretty little vesicle. It's not going to actually touch the host DNA. It's never going to make it into the nucleus. There's no transcriptional factors that are involved. And then we're going to digest it. We're going to present the antigen on the outside of the surface, right? That's what MHC2 molecules do, like macrophages, all right? They phagocytose and then they just spit out the antigens and put them on the surface of the MHC2 molecules, and then they continue with their job. The MHC1 to present antigen, the antigen actually has to be processed, okay? And that happens a lot with viruses. So keep this in mind. And we already know about activation here. Okay, so development and activity of lymphocytes. So essentially, when we're talking about B lymphocytes, right, we're talking about a little different process. So there's essentially two types of B cells. You have B1 cells and you have B2 cells. What do you need to know about B1 cells and B2 cells? So B1 cells possess a CD5 marker. That's what you need to know about it for identification purposes. Take into account, we talked about this before, when we use flow cytometry and monoclonal antibodies to identify CD markers on cell surfaces. And B2 cells, they account for most of the B lymphocytes in adults. So you can kind of think this in terms of maturity. B1 cells also have something to do with your NAbs. So we talked about natural antibodies or neutralizing antibodies last class. And it is hypothesized that the B1 cells are capable of producing these sort of neutralized antibodies that will eventually make their way genetically and then emerge from germ cell lines. And we did have a conversation about that, but it's not something that you need to commit to memory for the purpose of this class. So I want to get that straight. We have certain cell surface markers for B cells, just like we do for T cells and for every other type of cell in your body. So you're going to have an Ig receptor, right? That's going to bind to antigen. You're going to have the Fc receptor, right? FCR, sometimes it's called. That's going to specifically bind to the Fc portion of your antibody. Which type of antibody? Usually it's going to be IgG. All right. Also a complement receptor. It's your C3 complement receptor, which can bind to all of the subunits, activated or inactivated sort of versions of C3 and complement. And then your B cell surface antigens, which are going to be your major histocompatibility complexes, right? So your MHCs, what kind of MHC? It's going to be your MHC class 2. Why? Because B cells are capable of acting as antigen presenting cells. And remember that APCs are going to have class 2 MHCs. Okay, so this is really just one big thing that you have to get from this is that your B surface antigens, the major one is going to be your CD19. Okay, that's a huge identification marker for B lymphocytes. Like I said, when you're looking at these lymphocytes under a microscope, you're not going to say, hey, look, that's a B cell and that's a T cell and that's a helper T cell. Morphologically speaking, you're not going to be able to tell the difference. But through flow cytometry and monoclonal antibodies, you will be able to identify their specific and unique markers. So the CD19 is specific and unique to B lymphocytes, all right? And that makes sense because they're part of the immunoglobulin superfamily, right? And that's kind of what we associate our B cells with anyway. So when B cells are developing, they have precursors that have these delta chains, okay, in their cytoplasm. They do not have any immunoglobulin, meaning antibody, on their surface yet, okay, when we have these sort of primordial B cells. Like I'm talking just past that multipotent hematopoietic cell, right? It's only a stage or two after that. When they become more differentiated, but they're not fully differentiated, the B cells will develop cytoplasmic IgM antibody, okay, as well as surface. So you'll find the antibody inside the cytoplasm of the B cell, as well as on the surface of the B cell, but it's stuck there, okay? It's essentially a peripheral integrin, right? And then the mature B cells, okay, what they'll do is they'll eventually lose their cytoplasmic IgM antibody, and then they will add IgD to the surface of the IgM, okay? I'm going to kind of show you what that looks like in a second so that you aren't confused about it. So I'm actually going to take the opportunity, excuse me, to do that now. Let's start with a new page here. What they're saying is that as you have these B cells developing, they're going to lose and gain certain things, but we're talking about antigen specificity here, right? So you're not going to have, you have delta chains in here, you're not going to have any this B cell isn't capable of actually secreting or releasing any antibody right away, right? Because we started with that sort of primitive B cell. So you have these delta chains in the cytoplasm, but you don't have any antibody on the cell surface. Fine, doesn't matter. You don't need to know what the delta chains look like. It's not important at this stage in the game. As they differentiate further, what's going to happen? Well, when they differentiate further, they're going to start to produce this IgM inside the cell, as well as IgM, right? Although that's probably a bad picture because, oh, okay, sorry about that. So where we left off, so this sort of mature, not mature, immature, but a little more mature than this first guy. We have cytoplasmic IgM antibody, and then we also have surface cell or cell surface IgM on the antibody of this B cell. Remember, this is a B cell. Eventually, in the next stage, the IgM that's cytoplasmic is going to be lost. We don't need that anymore. We still have the IgM on the surface of the cell, okay? That stays there. But at this stage, eventually what's going to happen is we are going to add a new antibody called IgD, all right? What does IgD do? Well, that's important. IgD basically signals that this B cell is ready for activation. Ready for activation. Okay, it's ready to come into contact with its antigen. This is basically signaling the start of a naive B cell, right? And then it can go through clonal expansion after it's activated. It could make some memory cells. It could make some plasma cells. It could secrete antibody, and it could do its job and live out its lifelong dream. So that's cool. One thing I want to sort of mention here is that this process that's happening, this is a selection process, okay? This is part of B cell selection. And remember what we said before about T cell selection, that there's certain positive and negative selection steps. And what is the purpose of selection? It's essentially going to be the process of making sure that a B cell is doing its job, but also making sure that the B cell doesn't have any issues with it structurally, functionally, chemically. So at some point during this differentiation process, if this is selection, where is it happening? Well, it's a B cell. So this is going to be happening in the bone marrow, right? This is going to be happening in the bone marrow before the B cell is ever released into circulatory system. Now, what's happening in this step of selection is the IgM here, okay? The IgM is going to be checked. Remember I said selection process is almost like a quality control process, right? So this IgM is going to be checked. What is it going to be checked with? Well, this is essentially antibody receptor, right? It's going to be checked with self-antigens. Do we want our B cells to react and respond to self-antigens? We do not. So this is going to be checked. I'm not going to draw the whole thing out like I did with T cells because I think you get the idea now. But we're going to react our IgM on the cell surface of these B cells with self-antigen, and we're going to say, okay, what are we looking for? We're looking for some sort of negative selection, right? We do not want a reaction. Now, the cool thing about B cells that doesn't happen in T cells is that if there is a reaction, all right, let's say that there is a reaction with our self-antigens, these B cells are capable of making structural changes to their surface antibody receptors or the surface antibodies, all right, in the variable regions. So let's recall for a second that an antibody, okay, this isn't an IgM. Remember, IgM is a pentamer. It's a huge molecule. Let's say this is an IgG. So let's say if we're producing an IgG or whatnot, just for the shortcut version of it here. On the variable region of this antibody, we have the outer variable light chain, right, which could either be kappa or it can be lambda. And then our variable heavy chain can either be alpha or gamma, mu, epsilon, or delta, right? Okay. they're going to do switching, right? They're going to do this sort of class switching that happens until you get a combination of things here, all right, that do not react with the self-antigen. So that's really different in the B cell selection process, because they're able to say, hey, that doesn't work, take the B cell back, change some stuff on the actual antibody structure, and then test it again, okay? And when it tests, and then it's the right combination where it does not respond with self-antigen, okay, which is a good thing, then it gets to move on to this next step. And in the next step, IgD is going to be added. And when IgD is added to the cell surface, that's when this B cell is allowed to leave the bone marrow and go into the circulatory system, and then go to the lymph, whatever, wherever it's destined to go. All right, but it's ready to be activated. So it leaves as a naive B cell that's ready to be activated. Okay, but it does not leave the bone marrow, just like the T cells do not leave the thymus until they've gone through the selection process. All right, so just keep note that IgD means ready for activation. Okay. We are almost done here, kids. Thank you so very much. We have more about B cell activation. It's the same thing, except that you need to know that there are two different types of activation. There's specific and there's non-specific. So specific activation involves the antigen that is complementary to a particular Ig on the surface. So specific activation is sort of that classical activation that we think of when we think of B cell activation, but non-specific activation occurs with B cell mitogens. And a mitogen is essentially like a small peptide that induces mitosis, all right? So it induces cell proliferation, right? Cell division, cell growth. So B cells can be stimulated in their resting state to both enlarge, develop synthetic machinery, divide, mature, secrete antibody, all the jobs that we talked about that it does. Mitogens are able to and capable of activating these B cells in the resting state to do this. Okay, so plasma cell we know is just a differentiated B cell. It's a B cell that's been differentiated to secrete antibodies that are specific to whichever antigen it has come into contact with originally. So whichever antigen activated the B cell, essentially made it not naive anymore, that's the type of antibody that the antibodies will be specific to that are secreted from this plasma cell, right? We know that, we understand that. And also keep in mind that it is the end stage of the differentiation pathway. As far as we know, there's no further steps after becoming a plasma cell. Most of these plasma cells will sort of dissipate after they're not needed, but that's why during this process, B cells also differentiate not only into plasma cells, but into memory B cells, right? That are capable of lasting for a very long time, and they're capable of responding to antigen earlier in the second reinfection, and that is going to stimulate a faster, stronger immune response during second reinfection. And we had a whole picture about that. Okay, finally, just immune-mediated diseases, which we will get into eventually. Immunological disorders can be divided into primary processes, also acquired or secondary processes, all right? Keep in mind that acquired is something that you get that you're not born with, right? We talked about last class how congenital is something that you are born with. So a dysfunction in the immune organ itself, all right, and the immune organ in question is considered a primary disorder, okay? But in a secondary process, there's going to be some other disease that's happening, maybe not necessarily in the primary organ itself, that's going to affect the primary organ, right? And we're going to go through examples of what that means, but therapy can also give you this sort of secondary-like disease as well, because as we've talked about in the past, therapy is used for good things. There's pros to therapy, but there's usually consequences to long-term therapy as well. So diseases mediated through immune mechanisms can also be included in another category. So that means that, there's overlap here, which, like I said, is sort of the theme of this entire course: overlap, exceptions, caveats, whatnot. So we will get into these sort of immune-mediated diseases as time progresses. That's it for B cell and T cell development. I hope that it was helpful for you. And if for some reason you need any clarification on anything at all, please do not hesitate to reach out to me. A reminder, my office hours are 6 p.m. on Wednesdays. I'm always there for at least an hour. But if you come in before 7, I can stay longer with you, not a problem. You can also reach out to me to schedule a learning center, which is just a Zoom meeting on any other day besides my office hours. I will work around your schedule if I can. I will be communicating with you via Canvas through announcements, but everyone has access to my email. If you haven't seen it yet, it's under office hours on the Immuno Canvas course page. So reach out if you need anything, and I hope you guys have a good evening or a good morning or a good day or whatever time it is when you guys actually watch this. Okay, have a good one, guys.